In this video, we are going to be talking about point mutations. We were given this DNA sequence and we have to figure out which is the template strand and which is the coding strand. The start codon is ATG. By flipping the bottom strand to read 5' to 3', we can see that it also has an ATG start codon. The coding strand will have a stop codon 4 amino acids down from the start codon. The bottom strand contains methionine, which is the start codon, valine, serine, lysine, and then TAA, which will be UAA, and mRNA, which is a stop codon, so we know that the bottom strand is the coding strand. The mRNA sequence will be the same as the coding strand, but thymine will turn into uracil. By looking at the DNA and mRNA sequences without any mutations, we can see that the mRNA codes for methionine, valine, serine, and lysine. Point mutations can fall under two categories: a transition mutation or a transversion mutation. A transition is a mutation where the bases change from a purine to the other purine, or from a pyrimidine to the other pyrimidine. A transversion is a mutation when a purine base changes to a pyrimidine base, or when a pyrimidine base changes to a purine base. A missense mutation is a base change that changes the amino acid. This can change the function of the protein. The original DNA sequence has a T to an A change in the second codon, which is a transversion. GTC codes for valine, and the mutation will cause the mRNA to be GAC, which codes for aspartate. A nonsense mutation is one that causes a codon to become a stop codon. In this scenario, the fourth codon, AAG, becomes TAG, which is UAG, a stop codon in mRNA. This mutation is a transversion because A turns into T. This mutation will create a shorter protein and can cause the protein to function improperly or not at all. A silent mutation is a change in a codon that ends up coding for the same amino acid. In the third codon, C changes to T, which is a transition mutation because it changes from a pyrimidine to a pyrimidine. This is AGU and mRNA, which codes for serine, and so did the AGC. This mutation causes no function change, which is why it is called a silent mutation. A read-through mutation is a change in a stop codon that makes it code for an amino acid instead. This makes the protein longer and can alter the function. A change from a T to a G in the original stop codon will form a GAA codon in mRNA. This codes for glutamic acid, which is not a stop codon. Since the mutation is changing from a T to a G, the mutation is a transversion. Here are some tricks to help you remember this topic. A transition mutation is when a base changes to the other base from the same family. When we break this word up, transit means to move from one to the other, so we move from one pyrimidine or purine base to the other. A transversion mutation is when a base changes to another in a different family. When we break up the word, we have trans and version. Trans means the change, so we change the version of bases. You can remember missense by thinking of it as a miscommunication that changes the amino acid. Nonsense does not make sense for the protein, so it's going to stop coding. Silent mutations are quiet, so they are not noticed. Read-through mutations keep the amino acid sequence moving through instead of stopping.